All right, so the biological significance of those van der Waals interactions is that they are very easily broken, but they are reversible, or that breakage is reversible. Um, they can occur between any two atoms that are near one another. Now, where they are significant is in the stacking of DNA, as an example. So they are going to play a role, albeit maybe not the biggest one, in stabilizing biological macromolecules. So stacking of DNA and that double helix, whenever we're, just as we were talking about in the previous slide or previous chapter where we went from monomer up to that macromolecule, well, you will have van der Waals interactions in that macromolecular structure of DNA. Okay, the hydrophobic effect is maybe one of the more important parts about protein structure and types of interactions. I would say, yeah. So this refers to the association of interaction of nonpolar molecules or components of molecules in the aqueous solution. So it's one of the main factors behind protein folding, protein-protein association, also known as protein-protein interactions, and formation of lipid micelles, binding of steroid hormones to their receptors. And these do not arise because of some attractive direct force, but between two nonpolar molecules. What we have is, so ultimately we have something that is two molecules that are similar to one another, and they're not necessarily drawn to one another, but instead what we have is they're both in an environment that they don't particularly like, and they're kind of forced together. So it's not these strong forces that are attracting them, but instead strong forces that are basically trying to force them out of a situation or force them out of an environment. That's the hydrophobic effect. Okay. So the solubility of different gases and nonpolar solutes, what we have is nitrogen, oxygen, carbon dioxide. These are all nonpolar. And this goes back to your Gen Chem 1, where when you look at carbon dioxide, well, we've got these two opposite, we have these polar bonds, but they're opposite one another, so they're basically negating one another. Their solubility is going to be on the order of 0.018, so it's very insoluble, compared to ammonia or hydrogen sulfide. It's extremely soluble. That's going to go into solution very easily, and those are both polar substances. Okay, so what we're observing with that, with a non-polar substance compared to a polar substance, is water is going to surround those non-polar solutes, and that's going to give you a very low entropy compared to a high level of disorder where you have all these water molecules that are floating around one another. So low entropy is thermodynamically unfavorable. Thus, hydrophobic salts or solutes have very low solubility. So if there's some combination of solute and solvent that's going to be low entropy, it's also going to be low solubility. Okay. Now, the origin of that hydrophobic effect, this is kind of giving you a little bit of a display and a preview of compounds that we'll be talking about later this semester for exam 4 material, and that is your lipids. So lipid molecules disperse in the solution, and nonpolar tails of lipid molecules are surrounded by ordered water molecules. Entropy of that system is going to decrease. The system is now in an unfavorable state. So each lipid molecule forces all these surrounding H2O molecules to become highly ordered. So it's going to basically force them to position themselves in a way that they're kind of unfavorable or not as receptive to being in all those water molecules. Okay, so nonpolar portions of the amphipathic molecule aggregate so that fewer water molecules are ordered. So in that last slide, what we saw were these three individual lipids, and each one of those had a ton of water molecules surrounding them. So let's just say right here we've got 7, and then 1, 2, 3. I'm going to do this. There we go. we've got these three lipids independent of one another, and we've got a total of 21 water molecules that are a little bit more ordered because they've positioned themselves around that. That's a less favorable environment. What's going to happen is these three right here that I've drawn is that they're going to aggregate together, and it's going to look something like that. And when it looks like that, what's happened? Now, do we have water molecules? That's not really drawn how I want. We're going to have water molecules here or here? No, we're not. The only water molecules that we're going to have are going to be on the outside. And so that entropy is going to increase, so that disorder is going to increase, I'm sorry. And so that's a more favorable environment. So we don't have effectively 21 water molecules that have organized. Instead, we have only, 10 or something like that. So that's what we're observing here with this hydrophobic effect taking place. So all nonpolar groups are sequestered from the water, and the released water molecules increase the entropy further. Only the polar head groups are actually going to remain exposed. So with high enough concentration of amphipathic molecules, complete aggregation into micelles is actually going to be possible. So what we saw in the previous slide was we had, these three lipids that kind of aggregated together. Well, if you're in an environment where there are even more lipids, well, those can ultimately come together to make this 3D sphere. I know that's kind of redundant to say 3D sphere, but that's what we've got going on there. All right. Now, the hydrophobic effect favors ligand binding because binding sites of enzymes are often very hydrophobic. So those sites can bind hydrophobic substrates and ligands such as steroid hormones. So steroids, I know you probably talked about them in your Organic 1 class. And steroids are those 4 ring structures, lots of carbons and lots of hydrogens, fairly non-polar. So these steroid hormones, which displace water and increase entropy of the system, many drugs are designed to take advantage of that hydrophobic effect. So make a drug that's fairly hydrophobic, because what do you know the water is going to do? It's going to try to put it with a bunch of other hydrophobic stuff. Okay, so what we've got here is an example of that kind of being displayed. We've got a substrate that has all these water molecules surrounding it. Yeah, it's too ordered. It's too organized for water's preference. Well, this enzyme has that same sort of stuff going on. Well, if you can kind of force them together, what you can do is basically all these water molecules and all these water molecules, they can go off and just go bananas and do whatever they want. They can be all over here and everything like that. They can get separate from one another so that they're floating around doing whatever they want. So you've increased the entropy, you've increased the disorder of that system by doing that.